Hello, welcome to 13 Universal, and this is A-Level Caie Biology. We are now on Enzymes Topical Series 1 as we prepare towards the last paper 1 on May-June 2024 series. Okay, I'm wishing you the best. All right, this is schedule. Don't forget that the questions are all from past questions and masking of CIE examination. All right, let's go destroy some enzyme questions. Okay, get ready your writing material. Look at the first question. It says silk moth caterpillars have been genetically modified to produce a mixture of their own silk and such is the most stronger silk of the golden web spider. Okay, the spider silk polypeptide chain has many repeated sequences of two amino acids, glycine and alanine, arranged in crystalline structure. So you can see that. All right, so this question seems to be a question from biological molecular, but it's okay. Let's solve it. Okay, so this is the crystalline structure of this. Wow, it looks like that of diamond or whatever you call it. This is graphite. Okay, this is a biological molecule, right? This is a structure, right? So yes, what correctly describes the structure of spider silk? Pause the video and try it. What describes it? Okay, just pause the video. Look at question option A. Primary protein structure with regions of alpha helix. Now A is wrong because primary structure is the sequence of amino acids, all right, and it does not describe as alpha helix, so it doesn't show us amino acid. We don't see any amino acids, so it's not primary structure. All right, secondary structure proteins, all right, secondary protein structure with regions of beta pleated sheet. Yeah, that's correct. This secondary structure has alpha helix and beta pleated sheet. If it were alpha helix, guys, you know how alpha helix appears, right? It just is helical, just like this. All right, of course. Now the alpha helix, sorry, yes, alpha helix is helical, right? Yeah, but beta pleated sheet is zigzag, and the one we have on the body is zigzag. All right, so the answer actually is option, all right, B. So it's a secondary structure. Tertiary structure with regions of, no, tertiary is not tertiary structure. You can't see further folding, you can't see precise shape. Tertiary structure is precise 3D shape due to further folding. Okay, it's not showing. Quaternary structure shows more than one polypeptide chain. This is one polypeptide chain. In quaternary, it deals with more than one polypeptide chain, and it doesn't have beta pleated sheet. So the answer, guys, is A. All right, let's roll. Got a long way to go. Let's go. Which property of the tertiary structure of a globular protein enables it to catalyze metabolic reactions? Which property? So pause the video. You know the drills. Pause the video and solve the question. Okay, it says A. It has hydrophobic amino acid R group on the outside. No, that's not it. It's not a globular protein, because globular protein has hydrophilic R group on the outside, not hydrophobic. Hydrophilic R group on the outside, so that it can interact with water. That's why all globular proteins are soluble, because they have hydrophilic R group on the outside. But we're talking about enzyme now, right? So it said it will be denatured by high temperature. Now this has nothing to do with the globular protein, all right, how it enables it to catalyze metabolic reaction. So we're looking at how that structure, all right, gives the enzyme its catalytic activity, and B doesn't explain that. C says the R groups of some amino acids form bonds with substrate. Yeah, this explains it. Okay, all right, this explains it. You know that when substrate binds to the enzyme in its active site, let's assume that this is the enzyme. Is that okay? So this is the enzyme, this is the active site of the enzyme, so this is the substrate. Is that okay? Don't forget this is substrate S and this is enzyme E. So when it binds to this substrate, okay, this substrate to form ES complex, something that looks like this. Okay, trying to pardon me, I didn't get the diagram well. Let's erase it. Okay, and try again. Yeah, so it's going to give us ES complex. Remember, when enzyme binds to each substrate, it's going to give us ES complex. So something that looks like this. All right, so this ES complex. Now this ES complex is formed as a result of the bonds formed between the enzyme, right, and the substrate, right? Okay, so this ES complex, you can see, right? So just to explain this further, guys, you will see that this is the active site of the enzyme. So there are arrow groups of the amino acids in this active site, lining the active site. So there are amino acid lining the active site. So the amino acid lining the active site form temporal bond with substrate. Okay, and that actually can help, either exert strain to break the substrate, okay, or join the reactive group of substrate molecules to form a product. So C is the answer. Now this says the three-dimensional shape depends on hydrogen bonding. It doesn't depend on hydrogen bonding alone. It depends on, other bonds like disulfide bond, ionic bonding, and hydrophilic interaction. So C is our answer. Let's go. Students investigated the effect of substrate concentration on the activity of an enzyme. Wow. The graph shows the result of this investigation. Let's see the graph, guys. That's the graph. This is the activity. That's a dependent variable, always on y-axis. Okay, independent substrate concentration. All right. So what do they want us to do? So an increase in which factor could lead to a change in the activity of the enzyme at point X on the graph could lead to a change. Okay. So we have pH, substrate concentration, temperature. Which option is the correct option? So I want you to pause the video and then fry this fish because I am hungry. Okay, welcome back. Remember, the question says an increase in which factors could lead to a change in the activity of the enzyme. So what is it trying to say is that what can actually affect enzyme activity? What factors can affect enzyme activity here? Either increase or decrease it. Okay. Yeah, pH is one of the factors that affect enzyme activity. So if you increase the pH, right? So an increase in pH can change the activity. Either it increases the activity of the enzyme or decreases. Of course, substrate concentration, when increase it is going to cause an increase in the activity of the enzyme. Temperature can either increase or decrease it, depending. All right. So these three can actually cause a change in the activity because they are all factors that affect enzyme activity. All right. So the answer is C. Sorry, A. A is all of them. 1, 2. Okay. This is 3. Sorry, it's a typo. It's 3 here. So that's A. All right. Let's go to the next question. All right. It says the Michaelis-Menten constant Km is the substrate concentration. This is the definition of Km. This is the definition. It is the substrate concentration at which an enzyme works at half its maximum rate. Okay, so is enzyme concentration at half Vmax. Okay, that's Km. What is correct when Km is low? Yeah, what is correct when it is low, guys? Fry this fish. When Km is low, what is correct? Okay, pause the video and try to solve it. Now A says the enzyme has low affinity for the substrate concentration. All right, before I start solving question, I need to take you back because this is what dealing with critical thinking. But you cannot deal with critical thinking without, all right, memory. You need to know exactly the knowledge, all right, surrounding each concept. Don't forget that Km, all right, Km is indirectly proportional to enzyme affinity. Okay, so there's what called enzyme affinity, which is a tendency of an enzyme to combine with its substrate. We'll call it enzyme affinity. Enzyme affinity. So that means that when Km increases, the affinity of the enzyme decreases. So the enzyme binds, all right, less slow. Okay, binds less to its substrate. Okay. That means that Vmax also will be affected. So which option are aligns with this concept. Said if it has low, the value Km is low, it cannot have low affinity, because low Km will give us high affinity because of their relationship, inverse relationship. So A and B talks about low affinity. They are out of this game. They are disqualified. They can't be the answer because they don't align with the concept. Okay, let's come to C and D. The enzyme has high affinity, that's correct, for the substrate, and the quicker the reaction will proceed to its maximum. Okay, now D says the enzyme has high affinity for the substrate, all right, and the slower the reaction will proceed to it. Of course, if it has high affinity, the reaction will proceed quicker. It will work faster because high affinity means can binds more, so the rate of reaction should be high. So C is the answer. Okay. Number 5, typical enzymes are large globular proteins with specific tertiary shape. Which molecular interactions are directly involved in maintaining the tertiary structure? Which molecular interactions are directly involved in maintaining the tertiary shape? Okay, let's find out. But these are key things you have to look at. Okay, you can see them: hydrogen bonding, disulfide bridges, hydrophobic interactions. All right. So which one do you, all right, suggest, recommend? So pause the video and try it. Okay. So maintain tertiary structure. All of them are important. Okay, they help to maintain, right, the tertiary structure. Hydrogen bond, disulfide bridges. So all of them are involved. So the answer is A. Which statement about the Michaelis-Menten constant, all right, is correct for an enzyme with a low affinity for its substrate? Enzyme that has low affinity. Okay, so pause the video and check it. Remember our concept from the beginning. We said that Km, right, is what? Km value is indirectly proportional to enzyme affinity. I'll just write it, enzyme affinity. I'll write enzyme affinity as E, okay, A. EA, that's enzyme affinity for me. Okay, that Km is inversely proportional to enzyme affinity. So if we have low Km, guys, low affinity for each substrate, low, that means going to have high Km. So which option shows high Km? A says has high Km, all right. So A and B, all right, selected. C and D eliminated. Okay, because if it has low affinity, then Km should be high. Okay, let's examine A and B to find out the answer. It has high Km and reaches Vmax at high substrate concentration. Okay, B says it has high Km and reaches Vmax at low substrate concentration. All right, if it has high Km, that means that it will require high substrate concentration, right, to reach Vmax. Okay, let me explain this to you. Watch this. If you remember, guys, do you remember that graph of enzyme, graph of enzyme, of course, substrate concentration? This is subscope, all right. So substrate concentration against rate of reaction. Let me call it R here. You see, the graph usually goes up, right? Okay, and then levels. Okay. Now this is the first graph is without inhibitor. Second graph is with competing inhibitor. Can you see? Now let me use this to explain to you. Remember that both of them reach Vmax. The Vmax is the same. Okay, the Vmax is the same. All right. For both competitive, this is a graph of competitive. Yeah, that's C, right? And then this graph is a graph of without inhibitor. Okay, remember that Km, the formula of Km is half of Vmax. Substrate concentration at half Vmax. Substrate concentration at half Vmax. So that means you have to get your half Vmax. Now, which is likely here, right? This is half Vmax. That's Vmax over 2. Okay. Yeah, so when we trace it, okay, let's determine the Km for the reaction without without inhibitor, which is the first graph there, right? All right, so that's the first graph there. Yeah, so this is Km for without inhibitor. Now let's trace for with competitive inhibitor. If you see, can you see? Can you see that the Km value has increased when you introduce competitive inhibitor? The Km value increases. So all right, this is Km value for with competitive inhibitor. So I call it Km. KMC. Yeah, Km, right? C. You see that Km increase, all right, and because it increased by introduction of a competitive inhibitor, all right, what happens? So you see that this is Vmax. Can you see that this particular graph here, to reach Vmax, it requires higher substrate concentration, and because the Km is high, so the higher the Km, the higher the concentration. Look at it, the concentration required. This is Vmax, okay, the highest rate of reaction. All right, so it requires higher substrate concentration compared to this one that has low Km. This graph has low Km. Can you see it? So low Km requires low substrate concentration to reach Vmax. High Km requires high concentration of substrate to reach Vmax. Okay, so if you check A and B, you see why A, right? Because it has high Km, it's going to require high substrate concentration to reach, right, Vmax. And why does it have high Km? Because it has low affinity, and Km is inversely proportional to enzyme affinity. So the lower the affinity, the higher the Km, and the higher the Km, the higher the substrate concentration required, right, to reach Vmax. So that's why the answer is A, guys. Okay, it's A. Let's go to the next question. The graph shows the effect of an enzyme on a graph. This is a graph. Okay, so what do they want us to do? Which role identifies X, Y, and Z? So pause the video and try it. Okay, pause the video and try it. So you can see that X, right? You see that X requires higher energy for the reaction to occur. So X is uncatalyzed reaction. So you come to the options. Uncatalyzed is C and D, eliminates A and B. Okay, eliminates A and B. So now if we come to Y is catalyzed, so uncatalyzed, so catalyzed. Well, so we're working with C and D right now. Now Z, okay, of course you can check the energy, right? The energy content of the reaction. So this is saying overall energy released during the reaction. It's not, it's energy, all right, gained by the product. Now you can see, if you check this reaction, so energy is required for this reaction to occur. This is a transition state. Okay, so if you check, so you can see that uncatalyzed reaction, overall energy released during the reaction. So overall energy released during the reaction, because the product has a lower energy content, right, than the substrate. Do you get? So that's why, all right, this represents energy released, okay, during the reaction. All right, let's go to the next question, guys. The graph shows the results of two experiments on the effect of increasing substrate concentration on this rate of enzyme catalyzed reaction. You can see that one experiment was at a high concentration of enzyme, and the second was at a low concentration of enzyme. All other variables were standardized. So let's see the graph. Okay, so the question says, what would limit the initial rate of reaction at point X? What would limit it? What is the limiting factor? Right, pause the video and try to answer the question. What would limit this enzyme? Okay, all right, at X, what will limit it? Remember that the limiting factor here cannot be substrate concentration, because substrate concentration has reached its maximum value. Okay, now enzyme concentration, because we look at X here, high enzyme concentration, that's why it went up. So we're looking at what will limit it. All right, of course enzyme concentration seems to be close to the answer, but this substrate concentration cannot be the answer because it has reached its maximum value. Temperature and pH, we cannot say. Why? Because increase in pH, all right, may denature the enzyme, or decrease may denature the enzyme. The same thing with substrate, with temperature. So if you increase the temperature, it may denature the enzyme. Okay, if you increase the pH, it may denature the enzyme. But if we increase the enzyme concentration, you can see that it will increase the rate of reaction from this low, okay, from this Vmax here to a higher Vmax. So enzyme concentration should be the limiting factor there. It cannot be substrate because substrate has already reached the maximum. If you continue to increase it, it will remain constant. So enzyme concentration is the limiting factor. That is what will cause this reaction to go up. All right, which statement about Michaelis Menten constant of an enzyme? Okay, a correct statement. Oh yeah, so I wanted to fry this fish. I've said a lot about Michaelis Menten. All right, let's go. Number one says at Km value, at the Km value, half the active site of the enzyme should be occupied by the substrate. Is this correct? So make sure you pause the video and working on it. All right, at the Km value, half the active site of the enzyme should be occupied by the substrate. Yes. So that's Km is half of Vmax. That substrate concentration at half Vmax. Okay, Vmax is when the active sites are all full of the substrate. Okay, half Vmax means half of it. So number one is correct. 2 says Km represents the substrate concentration at which enzyme is working at half its maximum rate. Okay, that makes sense. Okay, 3 says the lower the Km value, the lower the affinity. Nope. The lower the Km value, the higher the affinity because they have inverse relationship. Okay, so 3 is wrong. 4 says when an enzyme has a high Km value, okay, the enzyme catalyzed reaction will proceed very slowly to its maximum. Yes, when it has a high Km value, that means it's going to have low affinity, so it's going to go slowly. So I think it's going to be 1. If you check what we have there, you can see that. Okay, 1 is correct, 2 is correct, and then 4. They're all correct. All right. So see this question. CYP3A4 is an important enzyme in human digestive system where it is needed to break down a range of different toxins. The activity of CYP3A4 has been shown to be reduced, okay, by a substance called furanocoumarins. Wow, what a name. Furanocoumarins are found in some fruits, and so dangerous concentration of toxins may develop in the human digestive system when fruits containing fumarocin are eaten. Okay, so remember the activity of this has been shown to reduce. And what is the role of this guy again? CYP3A4 is an important enzyme in human digestive system where it is needed to do what? To break down range of different toxins. Wow. Okay, from the information provided, guys, what can be concluded about molecules of enzyme CYP3A4? What a name, guys. All right, these are the options. Make your choice. So what can be concluded? Pause the video and solve it. That's how we roll it. Pause the video and solve it. Okay, A says they lower the activation energy of the toxin breakdown reactions. Yes, A is correct, because all enzymes lower activation energy. Okay, but why are B, C, D wrong? Okay, B said they bind specifically through the active site, right, to a substrate found in some fruits. And remember, they said it is needed to break down different toxins. Okay, the activity has been shown. Now they said that remember what they said here. It's an important enzyme in the digestive system, enzyme where it is needed. Okay, so it's an important enzyme in the human digestive system where it is needed to break down. And then, of course, look at what they said here. That fumaronic are found in some toxins, and so dangerous concentration may develop in the human, okay, when the food content are eaten. So this is said they bind specifically through the active site to a substrate. It does not bind to a substrate. It's involved with different toxins. Why different toxins? So the guy binds to many. So toxins are the substrate. Toxins. So that toxins as substrate has many toxins. It doesn't have one. Okay, that's why B is wrong. Now C says they change permanently when acted upon by fumarone. So we are not told whether fumarocoumarin is irreversible or competitive or non-competitive. Competitive are usually reversible in their activity, right? I mean inhibitors. Competitive inhibitors are usually reversible, but non-competitive reversible. We are not giving a clue to that, right? And also we are talking about this enzyme. All right, this is they resume normal activity when concentration of fumarate decreases. All right, we're not told that there is this talks about competitive, this talks about non-competitive. So none of this was connoted from the background information. So it's only A that sounds correct. All right, a fixed volume and concentration of substrate and enzyme were mixed, all other variables were kept constant. The enzyme catalyzed reaction was left until it was complete. Which graph shows how the rate of reaction changes with time? Okay. Yeah, the relationship between rate of reaction and time. So choose the option. So which option are you gonna go for? This A or B? Come on, C, D. Which one are you gonna get? Get one. Which one are you gonna get? Remember, whichever you're getting. Remember the concept. We don't do guesswork in OBJ. We follow the concept. Remember that rate of reaction, okay, is indirectly proportional to time. Don't forget that. This is a constant. Okay, so that means that the higher the rate, the shorter the time. So if the rate of reaction is high, the reaction occurs in a very short time. So which of these graphs shows us negative relationship between rate of reaction and time? Okay, this shows positive. This shows positive and neutral. This shows positive and negative. This shows only negative and of course on constant. So the rate of reaction has negative at the beginning. All right, so you can see that. So C, all right, shows the negative correlation. So as the reaction proceeds, rate of reaction, can you see rate of reaction increases? Increase means so rate of reaction decreases as reaction proceeds. That means at the beginning of the reaction, rate of reaction was very high. Okay, but as the time, all right, or reaction increases, the rate of reaction decreases. So C is the answer we're waiting for. Okay, so which region on the graph shows the activation energy of an enzyme catalyzed reaction? Don't waste time with this question. Okay, all right, they catalyze, all right, that's with enzyme. Enzyme is catalyzed. This is between the substrate, the reactant, okay, and the highest energy level. Do you get? So that means that B should be the activation energy. Okay, B should be activation energy needed, all right, for catalyzed reaction. That's enzyme. Enzyme is a catalyst. Okay. All right. So that's B. The graph shows the effect of an increasing substrate concentration on the rate of an enzyme catalyzed reaction. Right. You can see that this is maximum rate of reaction. This rate of reaction substrate. All right. This is P and Q substrate concentration here. Let's see what they got. Line P. This is information given to us. Line P represents the results when the enzyme is used at its optimum pH and optimum temperature. Wow, no wonder rate of reaction, okay, sorry, I begin. Optimum pH and optimum temperature and without an inhibitor. That's for P. Okay, line Q represents the result when the reaction conditions are changed. Okay, which descriptions of changes to the reaction conditions could result in line Q if all other conditions were kept constant? So which description will cause a decrease, right, from P to Q? Okay, has this kind of effect. Pause the video and change it. So look at all of this, 1, 2, 3. So which option do you choose, guy? Men and women, ladies and, okay, Man. Alright, boys and girls, which one do you choose? Alright, let's work on it. Pause the video and try to work on it. Number 1 says add an inhibitor that attaches to a site other than active site. Okay, this is a description of non-competing inhibitor. A non-competing inhibitor does not give this graph, this kind of graph. It doesn't give the graph of Q, right? Non-competing inhibitor does not reach, right, same Vmax, right? If you draw a graph of non-competing inhibitor, because this is without inhibitor, it goes like this, right? For non-competitive competitive inhibitor, it does not reach the same Vmax. Okay, so if I were to draw non-competitive inhibitor here, the reaction that shows it, maybe it goes like this, okay, and then levels, and then this number 1 describes it. Number 1, it says, so number 1 is wrong because Q doesn't show non-competitive inhibitor. Non-competitive inhibitor binds to allosteric site, which is any other site. That's what Q 1 is insinuating, that attaches to a site other than the active site. A site other than the active site is allosteric site. All right, so this describes non-competitive, but that graph does not show non-competitive. Q shows competitive. This is the one that shows competitive. Okay, so number 2 says add inhibitor that has similar shape to the substrate. Yeah, this is competitive inhibitor. All right, so number 2 is competitive inhibitor, and it actually aligns with graph Q. This is the graph for competitive inhibitor, and statement 2 describes, all right, competitive inhibitor because it has similar shape to the substrate, complementary to the active site. Okay, now number 3 says add an inhibitor, okay, that blocks the active site. Yes, that blocks the active, that's competitive. It binds to the active site. Number 4 says carry out the reaction at a higher temperature. If you carry out the reaction at a higher temperature, it's going to affect it. Okay, if you carry out at a higher temperature, of course, it's possible that it will denature the enzyme, and then the reaction will drop from this to this. High temperatures denature enzymes. Okay, all right. So 4 makes sense. So which option sounds correct? Oh, we have 2 and 3 only. Okay, why 2 and 3 only? Let's examine it. Okay, 2 and 3. 2 says inhibitor that has similar shape to the substrate. Yeah, this is correct. Okay, why is 4 wrong? Carry out the reaction at a high temperature. At a higher temperature. A temperature higher than the optimum. Alright, this is optimum. Okay, this is already up. If you carry it at higher than optimum, it will decrease. Sorry, I think there's a mistake here. 4 is correct. If you carry out a temperature, alright, reaction at a temperature higher than the optimum, alright, the rate of reaction will decrease. You get? So do you remember that graph? Okay, do you remember the graph? The graph of temperature. Alright, this is temperature and this is a reaction. Remember that as temperature increases, this is temperature. Let me use, this is temperature, TE, temperature. As temperature increases, rate of reaction increases until it reaches Vmax, the highest, and of course continues to decrease. So this is optimum temperature. So this is optimum. Optimum. So at temperatures higher, okay, than optimum temperatures, the rate of reaction will decrease. So this from here down is higher than the optimum here. So I think this is a typo error. So it should be 2, 3, and 4. The answer should be C. Okay, 2, 3, and 4 could actually affect this, right? Because we're looking at the descriptions that could cause changes. All right. So tell us what you think. Let's go to the next. The diagram shows a metabolic pathway. Let's see the metabolic pathway. Okay. They say what will be the effect of adding a small amount of a non-competitive inhibitor of an enzyme 2? So pause the video and try to solve the question. Pause it. Let's see what you can do. Okay. So what happens? what happens? What happens? Okay. If we add, remember what they said, what would be the effect of adding a small amount of non-competing inhibitor of enzyme 2? So if you add non-competing inhibitor of enzyme 2, small amount, okay, enzyme 2, so that means that some of the enzyme 2 will be, all right, inhibited. So that means the amount of enzyme 2 available to work on substrate X will be less, okay, because this enzyme 2 converts substance X to substance Y. So if you have an inhibitor inhibiting this and is non-competitive inhibitor, sometimes they denature the enzymes, okay. So that means some of these enzymes are going to be inhibited. And remember that enzyme 1 is working. Enzyme 1 is converting reactant to substance X, and then less of this is converted to this because only few enzymes are working on this, and this is increasing. That means that the amount of substance X will go high. So if you check A, enzyme 2 will be partially denatured. Well, not really, because some of them are working, all right. Enzyme B says substance X would increase in concentration. Yes, it will increase in concentration. Let's check C. Substance Y would no longer be formed. It will still be formed because not all enzymes here are denatured, or not all of them are inhibited. Let me use the word inhibited, all right. So some of them are still converting X to substance Y. So this is wrong. This initial reaction will no longer be metabolized. Initial reaction is reactant to substance X. It will still be metabolized because the enzyme 1 is working, it's not inhibited. So I think B is the answer there. All right. Okay, let's go on. 2 enzymes are added to a solution containing a low concentration of substrate, okay, that they both can use. I think that the word there, okay, that they can both use. So the both of them are using the substrate. So which statement is correct? Pause the video and try to solve it. Pause the video. Okay. 2 enzymes are added to a solution containing a low concentration of a substrate. So the substrate has a low concentration and both of them can use it. Then A says both enzymes will use equal amount of substrate. Well, it depends on the affinity for the substrate. So this is not always correct. Okay, it's not correct. B says neither enzyme will be able to use the substrate. They will use it. Okay. C says the enzyme with low Vmax will use less of the substrate than the other enzyme. Okay, is this correct or wrong? Then D says the enzyme with low Vmax will use more substrate than the other enzyme. Okay, remember that the enzyme with low Vmax, okay, is an enzyme, all right, the enzyme that has more likely less enzyme affinity. So that's why it's having low Vmax. Okay, it has less enzyme affinity, and if it has less enzyme affinity, okay, it's going to need more substrate to reach its Vmax. It's going to reach the same Vmax, right? So it's going to need more substrate, okay, to reach its Vmax, right? So low Vmax compared to substrate. So low Vmax means low, of course, low enzyme affinity. So it's going to need more substrate, okay? But if the enzyme has high Vmax, it requires less substrate to reach its Vmax, all right? So 16 says the enzyme trypsin hydrolyzes protein to amino acids. Trypsin does not function when it's very low at its 3D shape will be changed. Okay, because pH is very low. As its 3D shape will be changed. Okay, which explains this change in 3D shape if the pH is very low. They said that if the pH is very low, the 3D shape will change. It will change. So which explains this change in 3D? Okay. A says hydrogen ions attach themselves to negatively charged. Yes. Hydrogen ions are positively charged, so they bind to the negative charge R group and then break hydrogen bonds or ionic bonds, and that changes the 3D shape. Okay, so A is correct. What is wrong with B? Hydrogen ions disrupt the disulfide bond. It cannot disrupt the disulfide bond. It can only break hydrogen bonds or, okay, ionic bonds. Okay, and this is how it does it. The only thing that breaks disulfide bond is reducing agent. So C says hydrogen ions increase hydrogen bonding between amino. It doesn't increase it, it breaks it. Hydrogen ions reduces affinity of hydrophilic R group for water. This is not necessary. Okay, it's going to break the bond. So that's A. Okay, hydrogen ion breaks hydrogen bonds, and because low pH means higher concentration of hydrogen ions. Low pH, high concentration of hydrogen ions, all right, when they bind to the negative R group, and then they break ionic bonds and hydrogen bonds, and then the enzymes active site become distorted and less complementary. So A is correct. The flow chart shows some of the steps in the formation of collagen. You can see that. Let's walk fast. Triple peptide chain held together by X bond, right? So what's the question? Which row correctly identifies X, Y, and Z? So you remember collagen, okay? Collagen has, the molecule has triple peptide chains held together by hydrogen bonds. Yeah, H bonds. Okay, H bonds, not the sulfide. H bonds. So B and C selected, A and D eliminated. Triple helix. The triple helix, that forms triple helix. Okay, held together by intermolecular bonds to form fibril. So we don't use microfibril for collagen. What we use microfibril for is for cellulose, but for collagen we use fibril. So that's the answer. And then fibril forms fibers. So B is the answer. Right? Okay. Lysozyme occurs in human tears, saliva, milk, and mucus. Lysozyme has a role in the immune system and hydrolyzes peptidoglycan. Which description of lysozyme is correct, guys? Which one is correct? Right? So which answer is correct? Which description of lysozyme is correct? Of course, is extracellular enzyme, because it is secreted into tear, saliva, out of the cells into these body fluids, right? So this says extracellular, this says extracellular. B and D, okay, say intracellular, so we eliminate them. So we're working on A and C, okay? Now, it's not an antibody because it's not made by immune cells, okay? But it's antibacterial, right? Because it destroys peptidoglycan, all right, component of the cell wall. So it is C. The answer is C. It's against bacteria. Which letter in the flow chart shows the effect of adding a competitive inhibitor to enzyme catalyzed reaction? So I want you to quickly solve this, right? So of course, competitive inhibitor is reversible. It's a reversible reaction. Yes. Okay. Affects Vmax? No. It has the same Vmax. Okay. We've shown you the graph before. The Vmax is the same if you're using competitive inhibitor, reaches the Vmax, but at a higher concentration, right? So no, right? So B is the answer. Which statement describes some enzyme action? Some enzyme actions. Number one, see 1, 2, 3. Okay, let's see. So what's the answer, guys? Pause video and try it. Enzymes hold reacting molecules so that their reacting groups are close together. This is correct. Enzymes always hold reacting molecules so that their reacting groups are close together. This is correct about enzymes. Note it down. 2 says, in an enzyme catalyzed reaction, more molecules have sufficient energy to react than the absence. Yes, because enzymes lower activation energy. So more molecules will have more sufficient energy because the amount of energy required for that reaction is lowered. So enzymes subscribe, all right, the amount of energy required. So 2 is correct. 3 says reactions catalyzed by enzymes take place at a lower temperature than they would without the enzyme. Yes. Okay? Because the higher the energy required, the higher the temperature, right? So if we're going to need less energy because enzymes lower activation energy, therefore it's going to occur at a lower temperature, all right, than it should have occurred without the enzyme. So all the statements are correct, and that is why A is the correct option. Okay, lipase is an enzyme that catalyzes the hydrolysis of lipids, and an experiment was carried out to investigate changes in pH when lipase is added to a food sample containing a high proportion of lipids. High proportion of lipids. The results are shown in the graph. So these are the statements, right? It says which statements are possible explanations of the result of the experiment. You can see that. So I want you to solve the question. Tell us what the answer is. I have the solving the question. Okay. Tell us what the answer is. Tell us what the answer is. Okay. Tell us what it is. So guys, yeah, so which one is the answer? Because you can look at it. This is they said enzyme concentration becomes the limiting factor. Now they said which statements are possible explanations of the result of the experiment between 50 minutes and 60. Between 50 and 60. Okay. You can see that between 50 and 60 the pH of the sample, right, becomes constant, right? So what explains that? Enzyme concentration becomes the limiting factor. Now enzyme is not the limiting factor because we are the substrate, which is the lipid, right? Substrate concentration is not increasing. Substrate concentration is decreasing. Enzymes are breaking them down, and we're not supplying more substrate. Okay, so we're not supplying more substrate. All right, so you can see enzyme concentration cannot be the limiting factor. So the limiting factor should be substrate concentration. Yeah, because substrate is decreasing, so we're having less substrate. All right, as the reaction progresses, you can see as the reaction progresses, less substrate. Okay, more lipids are broken down to fatty acids and glycerol, and you see the fatty acid is what is causing the pH to decrease. Okay, all right, so number 2 should be there. 3 says all enzymes active sites are occupied. No, at this point, because substrate concentration is not being increased, substrate is being used up. In fact, at this point we have less substrate available. Okay, so all enzyme active sites are not being occupied, so 3 is not correct. 4 says denaturation of enzyme by the product. Yes, it's possible that enzyme is being denatured, because if you see the reaction started at pH 8. All right, the pH has changed from 8 to around 6.6 or thereabout. Okay, so it has moved from alkaline pH to acidic pH. Okay, so that means that fatty acid has been produced. Fatty acid is actually causing a decrease in the pH, so, and that could cause enzyme to denature, because this enzyme started at 8, so change in pH could break hydrogen bonds and ionic bonds. Is that okay? So 4 is correct. Now 5 says products are acting as competing inhibitor. Yeah, it's possible that that the products are acting as competing inhibitor. You know, there's what is called end product inhibitor, but you know, end product inhibitor is when we have excess product and then the product is now inhibiting the enzyme. So now, of course, 1 cannot be there, all right? 3 cannot be there. So any option that has 1 and 3, okay, like A and B, okay, and so you see the option is clear. Option is D, 2, 4, and 5. All right, let's quickly. So the painkiller ibuprofen reduces the activity of an enzyme involved in the production of prostaglandin. Ibuprofen binds reversibly to the active site of the enzyme. Consider. Which type of the enzyme inhibition describes this example and why? So if ibuprofen binds reversibly to active site, so which inhibitor? I want you to pause the video and answer the question. Okay, pause the video and answer the question. So yeah, which inhibitor binds reversibly to the active site? It binds to the active site. Now, of course, it cannot be non-competitive inhibitor. It's competitive. Do you get? It's competitive. It is competitive inhibitor. All right. Yeah, competitive inhibitor. All right. So it's A and B, but it said because ibuprofen alters the shape of that, it doesn't alter the shape of the active site. So altering the shape of active site is as a result of, right, non-competitive. So competitive, the inhibitors do not alter; they bind to the active site. So it is non-competitive that binds to allosteric site and then causes a change. But this says that B says competitive inhibitor, competitive inhibition because ibuprofen and substrate cannot bind at the same time. Yeah, they cannot bind at the same time because both of them are both complementary to the active site, the inhibitor, right, and the substrate. So if one is binding, the other one cannot bind. So B is the answer. Which description about all enzymes are correct? All enzymes. Okay, all enzymes. So solve this question. All right, solve the question. They catalyze breakdown of large molecules into small molecules. Not all enzymes catalyze the breakdown. Not all of them. Okay. Yeah. Because some of them join smaller molecules to form, right, large molecules. So this is okay, hydrolytic reaction. Some of them also carry out condensation reaction. So all enzymes do not do this. So this is wrong. Number 2 says all function inside the cell. Some enzymes function outside the cells. They are called extracellular. Right. So 1 and 2 out. 3 says form temporal bonds with substrate. Yeah, they form temporal bonds with substrate, which actually converts the substrate to product, either by splitting it or by joining it to another substrate. Have a tertiary structure. Yeah, all enzymes have tertiary structure. So it's 3 and 4. All right. Which graph correctly shows possible changes in energy levels as chemical reaction progresses with or without enzyme? With or without enzyme, right? Remember that the key says the thick line is with enzyme. The dotted line is without the enzyme. So pause the video and check it. Okay. Now enzymes lower activation energy. Okay. So you can see that. So and they should end up at the same energy level. So this and this are out of it. Okay. So and we're left with this and this. What's the difference between this and this? Because enzyme lowers activation energy. So and there is thick line. You can see. All right. If you check here, this should have a lower. All right. Activation energy. But this is telling us enzyme uses high activation energy. So it should be C. Okay, C. Alright, the graph shows a relationship between the concentration of substrate and the rate of enzyme catalyzed reaction. Alright, so you can see that this rate of enzyme catalyzed reaction, this concentration of substrate. Alright, which row shows how Km and Vmax for this reaction would be affected if the same reaction was carried out in the presence of a competitive inhibitor? Alright, so if you use a competitive inhibitor, I like you to pause this and try to solve it. So if you use a competitive inhibitor, how is it going to go? Of course, the graph is going to go like this. So it's going to have the same Vmax. Okay? If you check the Vmax is going to be the same. This Vmax. The same Vmax. But if you check, this is half Vmax. If you trace it, the Km, alright, without inhibitor will be this. Okay, that's here. All right, if you trace it to the second graph, the Km will be this. So the Km will increase, Vmax will be the same. So which option shows Km, right, increasing? So Km increases from here to here. Okay, Vmax is the same. So Km increases, Vmax, right, the same. That's option B. All right. So it says catechol is a chemical found in a number of fruits. Catechol can be oxidized to a quinone by the enzyme catechol oxidase. Catechol oxidase is inhibited. Look at it, it's inhibited by para-hydroxybenzoic acid, which is structurally similar. So what is the name of inhibitor that is similar to the substrate, which is catechol? Catechol is the substrate to catechol oxidase. PHBA is the inhibitor. All right. So and PHB has similar structure to catechol. What type of inhibitor is that? So in the presence of PHB, the Michaelis-Menten constant for catechol oxidase increases. All right, so it increases. So which inhibitor increases, of course, the Km value? All right, so I want you to, why is this showing? Okay, now look at it. How does PHB inhibit catechol oxidase? A says PHB and catechol can both bind to enzymes but not at the same time. Yes, because the PHB is is a competing inhibitor. Why? Because it's structurally similar to catechol, so they bind to the active site. Okay, so they bind to the active site. All right, so if you look at it, like, let me show you this, how this appears. So this is the enzyme, right? Okay, so this is the substrate, yeah, and this is, right, the inhibitor. Can you see that two of them are similar? So this is inhibitor, all right, I, this is substrate S, this is enzyme. So the substrate is catechol, you know that. Okay, why the inhibitor is PHB? So you see that A says PHB and catechol can both bind to the enzyme but not at the same time. Yeah, both of them are complementary. They cannot both bind to the enzyme. Okay, but they both can bind but not at the same time. So one is correct. PHB is a non-competing inhibitor. It's not a non-competing inhibitor. If it's a non-competing inhibitor, it will not be able to bind to the active site, but it can bind. Okay, so 2 is out. PHB 3 says decreases the Vmax of the reaction. Now, competing inhibitor does not decrease the Vmax. It's non-competing inhibitor, and PHB is a competing inhibitor. It has the same Vmax. So I think only one is correct. Okay, yeah, these 2 is for non-competing inhibitors. All right, let's look at the next question. The enzyme beta-galactosidase can catalyze the hydrolysis of 4 substrates with similar structures. So look at that. Each substrate gives a different Km value. For which substrate does B-galactosidase have the highest affinity? Okay, so remember, which one gives the highest affinity? Pause the video and try to solve the question. Okay, don't forget that Km, remember Km, right? Km is inversely proportional to enzyme affinity. Okay, so the one that has lowest Km will have the highest enzyme affinity. So the highest affinity will be the one that has the lowest Km. So which one has the lowest Km here? Which one has the lowest Km, guys? Okay. Look at the value. Okay. Shouldn't it be this? This is substrate 4. It has lowest Km, so it's going to have the highest, right, affinity. All right. An investigation was carried out on the effect of temperature on the activity of an enzyme. Okay, when it is immobilized and when it is non-mobilized, free in solution, the product of the enzyme catalyzed reaction causes a decrease in pH. All right, look at that. The graph shows the result of the investigation. So the question says, which would give the highest yield of product? Pause the video. Which will give us the highest yield of product? This is pH. Can you see? All right, as reaction is going on, pH is decreasing. Okay, time is increasing, pH is decreasing. All right, so which one gives us the highest yield of the product? So pause the video and try it. Which one gives us the highest yield of the product? Don't forget that rate of reaction, okay, all right, is inversely proportional to time. So, okay, and of course, as rate of reaction is increasing, what is happening, right, is that pH is also, because pH also increases. So pH is also increasing. So it seems that rate of reaction, all right, as rate of reaction increases, pH also decreases. So rate of reaction is also, if you look at it, because as rate of reaction, the reaction starts at high pH. So when the reaction continues, the pH starts decreasing. Okay, so that means that rate of reaction is also inversely proportional, right, with pH. So the one that gives us the lowest pH, okay, all right, will be the one that has the highest reaction. This is the one that gives us the lowest pH. This thick line guy, okay, that's the lowest pH, right? And that is non-immobilized 37 degrees Celsius. That is C. All right. So the graph shows the energy levels involved in an enzyme catalyzed reaction. Substrate molecules X and Y combine to give product Z. Which arrow shows the reduction in activation energy due to the enzyme? Reduction in activation energy. Okay, so this activation energy, this is catalyzed because it lowers the activation energy is here, but this lowers it. Okay, so initially it was from here to here. Okay, but this lowers it. So the difference, reduction is B. That is a B. So the difference between C and A is B. Okay, C minus A is B. Okay, so all right. So which graph correctly shows the activation energy of a reaction when an enzyme is added? Activation energy when an enzyme is added. Okay, so of course, when an enzyme is added, activation energy. So look at A. Why is A wrong? Pause the video and try it. Now remember that reactant must always be here. Okay. All right. Start with reactant. So you see, product is there. They show product here, which is wrong. So reactant should be here. Product should be here. So that is. So you see, reactant, and of course, product should also be at this side of the reaction. All right. So A and C wrong. If you check B, you see, look at B. This should be reactant, yeah, and this should be product. But mostly, the product have lower energy, all right, than the reactant. Okay, so this is reactant. Yeah, this is the product. Okay, so I think D is the answer. Okay, yeah, because the reaction starts with primary reactant. This lowers it, so energy is always released when you use catalyzed reaction. Okay, enzyme. So this is the product. This is reactant. This is activation energy. This is no enzyme. So product must always be here. Reactant must be here. So the energy, all right, value of reactant is usually greater than that of the product. Okay, so that's why D is the answer. So thank you guys for hanging on. Hope you enjoyed this class. Okay, this is Shedro. So see you. We're going to be revising cell membrane topicals. See you next video. Get ready for cell membrane. Bye.